Let's talk about cellular respiration. I'm going to be talking about the aerobic version of it. So what essentially happens is in cellular respiration, I recall that you have glucose reacting with oxygen. That's going to produce water, CO2, a lot of heat is going to be released, but most importantly, it produces ATP, which is essentially the energy source, right? Allows the cells, everything in your body to, be able to do stuff, right? Now, how is ATP an energy source? What essentially happens is you have ATP, right? Adenosine triphosphate. What essentially happens to that is this is going to react with water. And as a result, you're going to form adenosine diphosphate as well as a free phosphate group. All right. Now take a look at what happened. Adenosine triphosphate turned into adenosine diphosphate. So we essentially went from 3 phosphate groups into 2 phosphate groups. And then plus this one extra free phosphate, right? It's like one of the phosphate groups broke off from adenosine triphosphate, so it turned into adenosine diphosphate, and you just have this free phosphate group sort of lying around, right? And this reaction releases energy. All right, so this reaction releases energy, okay? And that is how ATP acts as an energy source. All right, what's going to happen is this water right over here, this is actually known as something called a hydrolysis reaction, but you're essentially using this water to sort of split this ATP apart. So this is how ATP looks like. You're going to have this group right over here, this group right over here, and this group right over here. This group is your adenine group. Adenine. This group is your ribose group. And these, of course, those are your phosphates, right? Those are your triphosphates, right? 1, 2, 3. Now, it turns out that at a pH of, around 7, what's going to happen is ATP is going to have a -4 charge or a 4- charge. And the reason for that is because, well, the phosphate groups, they are acidic, all right? And essentially what happens is these hydrogens that you see from the ATP group, they're going to deprotonate. So these hydrogens are going to get out of there, all right? So all of these hydrogens are going to be released. They're going to be released as H+ ions, all right? Remember that things that sort of release H+ ions, they're acidic, right? Because H+ ions are essentially what makes things acidic, right? Now, if you're removing a positive charge, well, what's going to happen is that the oxygen is now going to take on these sort of negative charges, right? So once all these hydrogens get out there, well, you're just going to be left with all these negative charges. So there we go. Now, what is going to happen is that this is going to react with water. So adenosine triphosphate, after the hydrogens have gone from it, what's going to happen is that you're going to have water. And the role of water in this is that water, what it wants to do is it wants to break this bond right over here. So it wants to break this bond right over here. It's going to try to break this bond. Okay. And you can see that if the water actually successfully breaks this bond, well, if I remove all of this right over here, notice how now we have adenosine diphosphate, right? Because there's only 2 phosphate groups. So if I were to break this bond right over here, I would be turning ATP into ADP, and that's exactly what I want because that releases energy, right? So that is my plan. Now, the problem with this is that, it really doesn't, occur if you don't have something known as ATPase, all right? This is an enzyme. All right, you need the help of this enzyme in order for this bond to be broken. The reason why is because we have all these negative charges, and remember that water has these lone pair electrons, right? Oxygen is very electronegative, so a negative charge meets a negative charge, and that's going to want to repel each other. So you're going to need the help of an enzyme, ATPase. What that's going to do is, well, ATPase is essentially going to have a lot of positive charges around it. So the positive charges basically draw these electrons towards it, and that allows the water, to do its thing and then finally break off this bond. All right. Now, what's going to happen here is imagine if this bond is broken, okay, and then the H2O sort of attaches to this. Now, what's going to happen is that one of the hydrogen atoms from this H2O molecule, it's going to sort of deprotonate. All right, it's going to turn into this H plus ion. So all you're left with is this OH minus. And if you can't sort of combine all of this together, this OH thing with this PO3 thingy right over here, well, now notice how you get a phosphorus, right? You're going to get 4 oxygens surrounding it, right, instead of 3, right? This is 3 oxygens. With this addition of the new oxygen, now you have 4 oxygens surrounding it, and it's going to be combined with another hydrogen atom. So this is actually going to turn into HPO4 with actually 2 minus charge. It actually has a 2 minus charge. And once again, HPO4 2 minus, this is something known as hydrogen phosphate, and it's also acidic. And what that means is that, once again, it can give away another one of its hydrogen ions. It can give another H plus charge away. And once another H plus charge away has been given away, right, what you're left with is, well, this is a phosphate group. This is a free phosphate group, right? PO4 3 minus. And, well, that kind of matched up with what I kind of said before, right? When you have ATP and you're reacting this with water, well, what's going to happen is, well, essentially, all the hydrogens, they're going to sort of be released as H plus ions, but what you're going to be left with is ADP, right? Because imagine ATP right over here, right? 3 phosphate groups. Imagine if this phosphate group sort of reacts and does its thing, how many phosphate groups are you left with? 2. So it's adenosine diphosphate, and you're going to be left with, well, take a look at here. You're going to be left with a free phosphate group here. It's important to understand that, well, cellular respiration, it's sort of like a cycle, right? From cellular respiration, you're forming ATP, right? And how do you form ATP? Well, what you do is you take ADP, right, adenosine diphosphate, and you're adding a free phosphate group, right, to form ATP. Now, when we need this energy source, right, when we need ATP in our lives, well, what happens is this ATP breaks down and it breaks back up into ADP. So this is a sort of cycle, right? We're sort of reusing these ADPs and these ATPs in order to either create energy or to use the energy whenever we need it. Glycolysis is the first sort of phase of cellular respiration, all right? And what happens there is you're taking glucose, right? And you're going to have this 2 NAD plus, all right? Now, this 2 NAD plus is very important, all right? It's going to be sort of carrying the energy amount, the amount of ATP we're going to generate in sort of the later steps of cellular respiration. Now, once again, this NAD plus right over here, so this NAD plus right over here, and you see this NADH right over here. Once again, it's going to be like this continuous cycle, just like we sort of seen with ADP and ATP. All right. Now, NAD plus to NADH. Well, what has to happen is, well, notice how there's an addition of hydrogen, right? So what's going to happen is you're going to grab a hydrogen ion, all right? And then what's going to happen is you're also going to grab 2 electrons, all right? You are going to grab 2 electrons. And all of this will be explained, in specific, where we get this sort of ingredients later. But just know that for right now, NAD plus reacts with this hydrogen ion, gains 2 electrons, and then it becomes NADH, right? Notice how there's 2 positive charges and then there's 2 negative charges, right? So all of that cancels out to make a neutral thing, all right? And what you'll see later is that this NADH will release these electrons and this hydrogen ion, and it's going to do some amazing stuff later. Now, glucose is a 6-carbon molecule. And what's going to happen essentially is it's going to be broken down into two 3-carbon molecules, right? So the 6-carbon molecule is going to be broken down into 2 3-carbon molecules, all right, which is this pyruvate right over here. And finally, you might have seen this, probably expected this, but finally you're going to have this 2 ADP. This is going to react with 2 free phosphates groups to form 2 ATP. It's actually important to note that this is actually the net sort of ATP. This is the net ATP. Because actually, in order for glycolysis to occur, you're actually going to have to input 2 ATPs, right? You're going to need 2 ATPs, and you're actually going to get out 4 ATPs. So the net is going to be 2 ATP, okay? Now let's get into the specifics of glycolysis, and let's actually see how all of this works, okay? So glycolysis is going to have 10 steps, right? It's going to have 10 steps, and there's going to be like an initial sort of investment, or like a sort of like um, initial sort of input of 2 ATPs during the start, and then after that you're going to get out 4 ATPs. So here are the steps in detail. So first of all, glucose is going to turn into glucose 6 phosphate. Now, what does that look like? Well, glucose has 6 carbons, right? That's why it's C6H12O6. And normally, it actually exists in a sort of ring, right? It exists in like a ring form. Now, at carbon 6, I mean, what's going to happen is that, well, attached to carbon 6 will now be a phosphate group, right? So this is why it's called glucose 6 phosphate, because, well, the phosphate is on the 6 glucose, right? Now, where do I get the phosphate from? Well, remember that this is the initial sort of investment phase. So in order for me to get this phosphate group, what I actually have to do is I actually have to take some ATP and I have to convert this into ADP and a free phosphate group. And this free phosphate group is now going to sort of drive this. All right. So it's going to allow me to sort of absorb this phosphate. All right. It's going to kind of give this phosphate to this glucose. Now, of course, you already know that you need enzymes. All right. So, I mean, it's good that we have the ingredients and everything, right? We have the phosphate, we have the glucose ready, but we need the enzyme for this reaction to happen. And the enzyme for this is hexokinase. Now, all these right over here are the respective enzymes. They're the enzymes for each sort of respective step. All right. Now, whenever you hear a kinase, you should be thinking of adding phosphate groups. All right. Whenever you hear kinase, that means that we're going to be adding phosphate groups. Okay. Now, the second step is that glucose 6 phosphate is going to turn into fructose 6 phosphate. And this is an isomerization reaction. Y'all know what isomers are, right? Isomers are essentially sort of molecules that have sort of the same sort of amount of atoms, right? But the thing is that they exist in sort of different forms, all right? Their structures are different. So for example, if I have, I'm just kind of making this up, but let's say if I have like a molecule like this, right? And then let's say if I have like a molecule like this, just making this up. But notice how we have the same amount of atoms and the same type of atoms in like each structure, but the actual structure itself is different, right? So these would be considered isomers of each other. Now this is actually pretty cool, but what happens is that glucose 6 phosphate, it's actually going to open up its ring. So this ring structure is going to open up, and essentially then isomerization occurs, right? And then what happens is that the ring closes back to form fructose 6 phosphate. Let's visualize how this looks. All right, this is pretty cool. So this is when glucose sort of gets out of its ring structure and gets into this linear structure, right? Now, so right over here, this is carbon 1, right? Notice how carbon 1 is bonded to 4 things, right? Remember, carbon wants to bond to 4 things, right? So this carbon number 1 is bonded to this oxygen, it's bonded to this hydrogen, it's bonded to this carbon right over here, and then it's bonded to this oxygen. Imagine if this happened, all right? Imagine instead of this bond with the oxygen to the carbon, imagine if this bond was an oxygen to this hydrogen. All right, so this bond did not exist. Imagine if this bond did not exist. Imagine instead of this sort of hydrogen bonded to the carbon, imagine if that bond did not exist. And imagine if this bond with this oxygen was a double bond. So notice how now we have this oxygen to hydrogen bond. We have this carbon to carbon bond that still stays intact. We have this carbon to sort of this oxygen bond right over here, this double bond right over here. And imagine if this carbon was actually also bonded to the hydrogen. So notice how this carbon still has 4 bonds, right? Which is our goal, right? We want to make carbon have 4 bonds. And here's how this would look like. So notice how this oxygen right over here is bonded to carbon number 5, right? This is carbon number 6. So this is carbon number 5 right over here, right? Notice how this carbon number 5 has an OH group now. Carbon number 5 has an OH group, right? And now notice how carbon number 1 now has a double bond to the oxygen and has a single bond to the hydrogen. Double bond to the oxygen, has a single bond to the hydrogen, and also has the bond to the next carbon. So that is essentially what happens when glucose opens up, right? The structure, the bonds, it changes a little bit. And yeah, that is sort of what happens. Now, how does this turn into fructose? Fructose 6 phosphate. Okay, so I want you to imagine this, all right? Imagine if this hydrogen went up here to bond with this oxygen. So imagine if this hydrogen right over here went up to bond with this oxygen. So that means that this oxygen to carbon bond, this is a single bond now. Because remember, oxygen wants to make 2 bonds. So if I'm making a new bond with right over here, then I have to sort of lose a bond there. So I sort of have to lose a bond here, right? And now what's going to happen is imagine if this hydrogen right over here bonds with this carbon, right? Because now this carbon's unhappy. Remember, carbon wants to make 4 bonds. So now this carbon's unhappy. So let's give it a hydrogen, right? So imagine if I get rid of this hydrogen, and now I just have this hydrogen right over here bonded to this carbon. But then now this carbon is unhappy because it only has 3 bonds. So let's make this carbon and this oxygen a double bond. Ooh, it looks like everybody's happy now, right? So what we essentially do is we kind of moved all the hydrogens up, and then now we have this new double bond with the oxygen. And that is essentially what happens with this isomerization. Sorry, so this is fructose 6-phosphate in its open chain form, and it's going to close back up, and then there it is. This is going to be my fructose. Sorry, not that one. Fructose 6-phosphate. All right. Now we're going to invest another ATP. So right, we invested 1 ATP in the beginning. Now let's invest another ATP. We're going to turn fructose 6-phosphate into fructose 1,6-bisphosphate. So now there's going to be 2 phosphate groups, right? There's going to be 1 phosphate group on carbon 1, and there's going to be 1 phosphate group on carbon 6, right? Hence why it's 1,6, and then it's bisphosphate, right? It's bisphosphate because, well, these phosphates are, sort of away from each other, not sort of connected, right? They're on these different carbons, right? They're all the way apart from each other. So that's bisphosphate. And this is going to be a single arrow. And that just means that, well, it's not reversible. So once I turn fructose 6-phosphate into fructose 1,6-bisphosphate, I can't go back. All right. Once you go there, you cannot go back. And once again, if you need sort of the name of the enzyme that powers up this step, well, once again, you have this right over here. Notice how, once again, it's a kinase, right? Phosphofructokinase, because, well, we're adding a phosphate group. So once again, for fructose 6-phosphate to fructose 1,6-bisphosphate, we have to add a phosphate group, right? And where does this phosphate group come from? It comes from converting ATP to ADP. So we have invested another one of our ATPs. So far we have invested 2 ATPs. And I promise you guys that we got 4 ATPs back with a net gain of 2 ATPs. So let's see, let's kind of watch this process happen. Now, the fructose 1,6-bisphosphate, well, it's going to break up into 2 things. And I really like this diagram because, well, it shows kind of the 2 things that it has, right? And it breaks up into glyceraldehyde 3-phosphate, right? 3-phosphate because, well, it's located on carbon 3, right? And it's going to break up into dihydroxyacetone. All right, I always want to make sure that I pronounce that correct. And phosphate. Okay, so it's going to break up into these 2 parts, right? We're essentially breaking up our 6-carbon atom into 2 3-carbon atoms, right? As you can see. Now, here is the payoff phase. All right, so we have the investment sort of phase. Now this is where we get our ATPs. All right, this is where we get our ATPs. So what's going to happen is that this glyceraldehyde 3-phosphate and this dihydroxyacetone phosphate, all right, it's going to turn into glyceraldehyde 3-phosphate. It's going to turn into 2 of these, okay? How does that work? Well, what essentially happens is, well, we already have 1 glyceraldehyde 3-phosphate, and we're somehow turning both of these into 2 glyceraldehyde 3-phosphates. How do we do this? This is glyceraldehyde 3-phosphate right over here. And this is the other thing that we want to turn into this, right? We want to turn somehow the pink thing into the blue thing. How do we do this? Imagine if I took one of the hydrogens here and I added it to this oxygen. So now I added a hydrogen here. I erased this 2 right over here. And this right over here, because oxygen wants to make 2 bonds only, it's bonded to this hydrogen. So that means it only has to be bonded once to this carbon right over here. So this double bond is just going to turn into a single bond right over here. All right. Now what happens? Well, the next thing that sort of happens is, well, this hydrogen right over here, because this carbon is not happy, right? Now this carbon, instead of 4 bonds, it only has 3 bonds, right? So this hydrogen is going to help it out, right? What this hydrogen is going to do is it's sort of going to bond to this carbon. So this hydrogen right over here is going to bond to this carbon. Hence why there's a CH here, right? And finally, this carbon is going to be double bonded to the oxygen, and then it's going to be single bonded to the hydrogen. And boom, there we go. This is how this pink thing converts into this blue thing. Okay, so there we have that. All right, so we're gonna have 2 glyceraldehyde 3-phosphates here. Now here's where it gets like a little bit more complicated. So this is gonna involve another phosphate, inorganic phosphate group. How this phosphate right over here is actually it actually doesn't come from the ATP breaking down. It's actually inorganic phosphate that's already dissolved in the sort of cytosol. So it already sort of exists in your cells naturally, right? So these inorganic phosphates do not require ATP to be sort of added. All right. Now, what's going to happen is recall that originally we had a 6-carbon molecule, and now we have two. We have 2 3-carbon molecules. So it's important to note that this reaction will actually happen sort of twice, right? Because now we have 2 3-carbon molecules. So this is why there's a 2 in front of everything, right? It just shows that, well, this reaction is going to be happening 2 times, right? Because we have 2 3-carbon molecules, right? We've broken it down. So in this case, this phosphate, well, notice here, this is our original structure. Now what has happened? Instead of this hydrogen here, we have replaced it with this sort of phosphate right over here, right? So this is going to become glyceraldehyde 3-phosphate is now going to become 1,3-bis, right? Bis, because now we have 2 phosphates now in our molecule, bisphosphate glycerate. All right. Now, what's actually going to happen is this is actually going to release 2 electrons. So this carbon right over here is actually going to release 2 electrons, and it's actually also going to release this hydrogen ion, right? It's actually going to also release a hydrogen ion, right? And remember what we said, right? If we have NAD plus, right? If we have NAD plus and we react this with a hydrogen ion and this sort of 2 electrons, well, what do we get? We get NADH, which is what we have right over here. This reaction happens 2 times, so it's multiplied by 2. So the net gain from this is 2 NADH. But all right, you kind of see the vision here, right? The carbon is going to lose 2 electrons. It's going to lose this hydrogen ion. So then you're going to have this NADH that's going to be formed from all of this. Okay. Now it's important to point out some vocabulary terms. So notice how this glyceraldehyde 3-phosphate right over here, right? It's losing electrons, right? This right over here is losing electrons, right? This essentially it's giving away these electrons right over here to the NAD plus, and this NAD plus is picking up those electrons, and this is forming NADH. So we would say that G3P essentially this is being oxidized, meaning that we are going to be losing electrons, right? This is giving away the electrons. Now, if something is giving away these electrons, something has to be receiving them, right? And so this NAD plus is receiving the electrons, and therefore this is a reduction reaction. We would call this a reduction reaction. All right, so I've seen a lot of people do this. So Leo the lion goes grr. Leo the lion goes grr. Loss of electrons is oxidation. Gain of electrons is reduction, right? So that's like a good mnemonic, or just a good phrase in order to memorize all of this. All right, so that's going to be formed. Now we are on to step 7. All right. Now we're on to step 7. So what's going to happen? We've added we added this phosphate, right? From step 6, we added this phosphate to our molecule so that we formed this, right? Now what's going to happen is, well, this phosphate group that we're going to add, well, it's going to sort of break down. Now we're going to start creating some ATP. So this phosphate right over here is going to break down. It's actually going to combine with an adenosine diphosphate, and once again, it's going to form an adenosine triphosphate. And because this reaction happens twice, we're going to get 2 ATPs out of this. All right, a net gain of, not a net gain, but a total of 2 ATPs that are being formed in this step. Okay, so this phosphate sort of detaches from all of this. All right. Now we are on. Now after this is step 7, so we are forming 3-phosphoglycerate. Now we are on step 8. Okay. Step 8. I like to look at this as the hydrogen and the phosphate switching step. All right. This is the switching step, because what essentially happens is, well, imagine if this hydrogen swapped with this phosphate group. So now we're going to get this, right? Now instead of O here, we're going to have a O bonded to a hydrogen. And then now instead of just OH here, that's going to switch, right? It's going to form an OP, right? So this is just a switching step where the sort of phosphate and everything here goes from the carbon 3 to now carbon 2, right? Everything's sort of swapped, right? Now we are on step 9, and once again, it's pretty good to pay attention to all of these enzymes that are taking place here. Oh, I say taking place. All the enzymes that are sort of involved in this reaction. So, all right, now we're on step 9. All right, step 9 occurs when, okay, let me take this hydrogen now, and let me take this OH out. When I have hydrogen and I have this OH, what am I forming? I'm forming H2O. And H2O, remember, H2O is, well, a byproduct. It's a product of cellular respiration. So there we go. This is where we get the H2O from. Step 9 is where we get the H2O from. Once again, it's multiplied by 2 because we have 2 of these reactions forming. And finally, we are going to lose our phosphate group. So this phosphate group is going to detach, right? And this is actually going to form ATP because remember, right? ADP, if we get rid of this phosphate right over here and we add that to ADP, we get 2 ATP. So we have gained another 2 ATP here, all right? So our net gain is 2 ATPs, right? We needed 2 ATPs at the start, but then we gained 4 ATPs, so that's going to be a net of 2 ATPs. And another important thing that we're going to form is something called pyruvate, all right? Now, pyruvate is this right over here. It's going to be this right over here. It's a little different. It's a little different from what's up here because what we have essentially done is we take this double bond right over here and we said, okay, if I want this to be a single bond, well, then what do I have to do? I have to add hydrogen here, right? So this becomes a CH3. So this is a single bond now, right? And now, instead of this carbon and this oxygen bonded to this phosphate and everything, this carbon is now going to be a double bond with this oxygen. So now it's going to be a double bond with this oxygen because this oxygen has lost the phosphate group. So now oxygen only has one bond. It's unhappy. So we're going to form this double bond here. And then everything else stays the same. And once again, you might be asking, where does all of this sort of hydrogen be coming from, right? How do I get from CH2 to CH3? And my answer to that is, well, the hydrogen needed for this is just freely available in the cytosol. So yeah, cells are, once again, cells are full of water and they're full of ions. So that includes like protons. So yeah, you just grab a proton from the cytosol. So for glycolysis, remember, we form 2 pyruvate, 2 NADH, and a net gain of 2 ATP. Now after the glycolysis is something known as pyruvate oxidation. There is a problem though. Glycolysis occurs in the cytoplasm. However, pyruvate oxidation occurs in the mitochondria matrix. So those are 2 different locations, meaning that we're going to need to transport stuff. All right. And this is where it gets a little bit confusing. So lock in. So essentially, right, in the mitochondria, you have an outer membrane. You have an inner membrane. Now the space that's in between the outer membrane and the inner membrane is called the intermembrane space, and you can kind of see why it's called that. And then inside the inner membrane, we have the matrix. So what I have here is I have pyruvate, and I'm also going to have some NADH, and I want to transport them inside the matrix. So how do I do this? Well, it turns out that pyruvate can actually enter it through something known as pyruvate. Oh my god. Pyruvate, and then it's called transcolase. This is going to help facilitate the transportation from the intermembrane space to the inside. So inside the matrix, right? I want to get from the intermembrane space inside the matrix. So I have to pass through the inner membrane, and pyruvate transcolase helps me with that. Now, NADH is a little bit more complicated than that. But essentially, to kind of simplify things, essentially what happens is NADH actually turns into NAD plus, it's going to release a hydrogen ion, and it's going to have these 2 electrons. And what's going to happen is these 2 electrons are actually the ones who are moving inside. All right. So NADH actually does not go inside the mitochondria matrix. However, the electrons from NADH do go inside. All right. Now you might be wondering, okay, that's pretty cool, but what does this NADH do, and what does this H plus ion do? Well, they stay in the cytoplasm. And NAD plus, remember, this is a cycle, right? Remember, we're always cycling back from NAD plus to NADH, right? It's a constant cycle. So the fact that we're producing this NAD plus and it's staying inside the cytoplasm means that cellular respiration is going to keep on happening, right? So that's a good thing. Now, these two electrons, they can travel through, they have two paths to travel through. All right. And depending on the path, well, that's going to produce different amounts of energy. So depending on which path you take, that's going to lead to a different amount of energy yield. And that's why actually cellular respiration, has like a range of ATP values. Like usually when you see sort of the cellular respiration equation written out, you'll actually see a net yield of 30 to 38 ATPs. And the reason why it's a range depends on which path you take here. You can either take the moly aspartate shuttle. All right, think about like a train, right? So like a freaking shuttle, and then these electrons can travel through there. And when the electrons travel through there, what's essentially going to happen is NADH plus is going to turn into NADH. All right. So if you take the moly aspartate shuttle, well, that's gonna essentially reduce NADH, right? Because you're gaining electrons, right? NADH is going to gain these two electrons, and then as well as a hydrogen ion, and it's going to become NADH. And when this happens, when you use the moly aspartate shuttle, this is going to yield around 3 ATP. So sort of every sort of NADH that's inside the mitochondrial matrix, right? Because there's a difference, right? This NADH is outside the matrix, right? It's in the cytoplasm. This NADH is inside the matrix, right? So there's a sort of difference there, right? And this NADH, it's just going to give away its 2 electrons, and it's going to run away inside the cytoplasm. However, these 2 electrons are going to be able to reduce NAD plus that's already in the matrix, right? There's stuff already. There's this NADH stuff that's already inside the sort of matrix, so it doesn't have to pass this barrier, right? Now, if you take the glycerol 3-phosphate shuttle, you take this shuttle right over here. Oh, what's going to happen? This is actually going to reduce something known as FAD, and it's going to reduce it into FADH₂. So in order for this reduction to happen, right, because we're gaining 2 electrons, well, we're also gaining 2 protons as well. So if we're taking the glycerol 3-phosphate path, you're reducing FAD into FADH₂, and every FADH₂ that you have, that's going to have a yield of around 2 ATPs. So depending on the path you take, that's going to lead to a different amount of ATP yield, and that's going to lead to overall different yield for the total reaction. Now, the type of path that you take depends on the type of tissue you're talking about. So yeah, that's where you sort of can differentiate between the 2 paths. All right, so now let's say we're inside the matrix. All right, we are inside our matrix. And inside our matrix, remember, we have our 2 NADH, actually 2 pyruvate. We have transported our pyruvate inside the mitochondria matrix. And let's just say, for simplicity's sake, I have 2 NADH. All right, I have 2 NADH to the side. All right, let's just say. Now, for pyruvate oxidation, NADH, this NADH right over here isn't actually going to be participating in our reaction, but this is just written over here to keep in mind that this is the amount of NADH that we have inside our mitochondrial matrix. So pyruvate is actually going to react with something known as coenzyme A. A coenzyme is just sort of a non-protein. It's a non-protein sort of, pretty sure, organic molecule, so a non-protein organic molecule. And what it does is it's sort of like a helper for a regular enzyme, right? Coenzyme, it goes with the enzyme, right? It's a helper for an enzyme. So imagine, let's say if this is like a chemical bond that I want to break, right? And enzymes can help with breaking the chemical bonds, right? So the enzyme can help break up the chemical bond, right? And let's just say these 2 molecules try and go inside the enzyme. Okay, this enzyme is not really in the correct shape to do this. So what happens is you can have a coenzyme. And what the coenzyme does is, well, it kind of fixes that. So it kind fixes that. Let me actually do a better drawing of this. How would this look? Oh yeah, look like this. Something like this. So this would be the coenzyme right over here. And now when I place my compound inside, well, now the bond breaking has been assisted by my coenzyme. So it just helps with like the shape of the enzyme and everything. So pyruvate is going to react with coenzyme A to form acetyl coenzyme A, and it's actually going to release CO2. So this is the step where CO2 is going to be released. In addition, you're going to have NADH, not NADH, NAD plus, because remember, this is something known as pyruvate oxidation, right? So what's happening? Pyruvate is being oxidized. It's losing electrons. So something has to gain those electrons. So we're going to have an NAD plus, and that's going to gain two of the electrons, and that's going to form NADH. All right. Well, that's going to gain NADH. Now, because we have two pyruvates, well, we're going to gain two NADHs, and we're going to gain two of everything right over here. So if I gain two NADH, that's going to add on an additional two NADH to what I originally have. So now I have 4 NADH after all of this. So sort of let's visualize what's happening here. So right now, this is pyruvate. And what's essentially happening here is we are removing this CO2 right over here, right? This is sort of CO2, and essentially we're replacing that with sulfur and the coenzyme A part. So that's the sort of coenzyme A part. So actually, this is something known as a link reaction. This is also known as the link reaction. And this is also something known as decarboxylation. And this is because a carboxyl group is removed from pyruvate. So for carboxylic acids, you might remember that it's COOH, right? You have a carbon double bonded to oxygen, then single bonded to another oxygen, and then it has a hydrogen attached to it. But in this case, pyruvate actually does not have this hydrogen attached to it, and instead this oxygen has a negative charge. And yeah, this is just because, like, imagine if you have a carboxylic acid, like imagine if pyruvate still had a hydrogen attached to it, but then once again, carboxylic acids, they're acids, right? They lose their hydrogen ion. So this H plus ion deprotonates, right? You have an H plus ion that goes away from it. So then this oxygen gains a negative sort of charge, right? It has this negative charge. And to simplify things, we just call it a sort of carboxyl group, but it's actually a carboxylate ion. So that's like kind of like the proper term for it, but we just call it a carboxyl sort of decarboxylation for this sort of reaction, right? Because we're grabbing this CO2 group right over here, essentially, and yeah, we're creating CO2 on our product side. So that is where the CO2 comes from. Yeah, we also have to add our coenzyme A, but yeah. And just so y'all know, the sulfur right over here comes from the coenzyme A group. So yeah, so everything here is fine, right? We have this is sort of balanced out, this is sort of balanced out, and then that is sort of balanced out as well. So that is pyruvate oxidation here. Now, once again, right, you're giving away the electrons, right? So don't forget that if this is the oxidation part, then what happens is that NADH, right? NAD plus, right? It's going to gain 2 electrons as well as a hydrogen ion, right? The 2 electrons basically come from, well, the oxidation, right? Where the where the pyruvate essentially loses 2 electrons when they're trying to combine with this coenzyme A, and this is going to form NADH. So there we go. And remember that because we have 2 pyruvates, once again, all of this is doubled. So in total, we have 2 NADHs here, 2 NADHs there. So we have 4 NADHs in total. We have this acetyl coenzyme A, and we have the CO2 that's released, which we get out of our body, right? We breathe out CO2. So yeah, there we go. Now, after sort of everything here, after we have finished our pyruvate oxidation, we then move on to the Krebs cycle. And this also occurs in the mitochondrial matrix. After the Krebs cycle, then we have the electron transport chain. And then after the electron transport chain, we have something, we're going to explore the proton gradient thing. All right. There's a proton gradient thing. It's pretty cool. And at the end, we'll see how diffusion can actually make ATP. So right now we are going to be on the Krebs cycle. This also takes place in the matrix. Let's look at it. So after your pyruvate oxidation, well, you're gonna go to the Krebs cycle. Now it's important to note that the Krebs cycle is only for acetyl-CoA. All right. So when you form this acetyl-CoA, it's gonna go directly to the Krebs cycle. NADH is actually stored so that it goes from the pyruvate oxidation and it goes straight to the electron transport chain. So the pyruvate, so the NADH actually skips the Krebs cycle. It's not involved in it. It's only the acetyl-CoA that is going to be involved in it. Now I'm really just going to be focusing on the steps that actually do stuff. So first of all, you're going to have acetyl-CoA, and what's actually going to happen is the coenzyme is actually going to detach from it. So for kind of step one, right, what's going to happen is you're going to have an oxaloacetate, and that's a carbon, that's a 4 carbon molecule. What's going to happen is that's going to react with acetyl-CoA, and that's going to form citrate. Now, notice how citrate does not have the coenzyme A on it. So essentially, the coenzyme A, it's going to dip, right? So it's going to dip for the first step of the sort of Krebs cycle. Well, it's not really a first step because remember, it's a cycle. This thing is going to keep on going, but you know that's just the way it goes. And then after that, it's going to turn to isocitrate. The reason why it's called isocitrate is because this is an isomer of citrate. So isocitrate is an isomer of the regular citrate. What essentially happens is, imagine if you move this oxygen down one. So we move the oxygen down. Well, now we're just going to be left with a hydrogen and carbon bond here, and then this oxygen is just going to be moved down one. All right. Now, after this is where it gets interesting. So after we form isocitrate, then we're going to form this compound. Now, this symbol right over here is just alpha symbol. So this is going to be alpha ketoglutarate. And during this step, right, when we go from isocitrate to alpha ketoglutarate, we're going to produce one NADH, and we're also going to produce CO2. Notice how we have essentially lost this CO2 group right over here. And we also, it also looks like we have lost a hydrogen. So if you were to take a look at isocitrate, look at this, and then now look at alpha ketoglutarate, right? So now we only have 5 carbons left because one of it left as CO2. And you also notice how one of the protons sort of disappeared. And it looks like this hydrogen atom right over here sort of bumped up to this spot, right? So now we have CH2 here, right? One of the hydrogens sort of bumped up to this spot. And now we have this carbon double bond of this oxygen now. So that looks like, that looks good. Now the CoA, the coenzyme A, is actually going to come back into the reaction here, and from alpha ketoglutarate to succinyl-CoA, we are actually going to release another CO2, and we're actually going to gain another NADH. All right, so, and there we go. Now from succinyl-CoA to succinate, right over here. Well, what has happened? Well, it looks like the CoA, the coenzyme A, has gone out of the way, and well, from succinyl-CoA to succinate, what has happened is we produced something. I'm actually going to produce something known as GTP. This sounds very similar to ATP. GTP is just ATP, but imagine instead of the adenine, sort of the adenine part of the molecule, imagine if that was replaced. With a sort of guanine base. So it has a guanine base, hence why it starts out with a G instead of a A. Now, GTP can be converted into ATP. So if we have GTP, what we do is we have ADP, and this yields ATP and GDP. So you kind of see what I did here, right? This is triphosphate. So what happens is GTP gives one of its phosphate groups to ADP, so now it becomes ATP. However, because GTP lost one of its phosphate groups, then it becomes GDP. All right, so it's kind of like they just kind of swap sort of phosphate groups, right? But you know, at the end of the day, we're gonna produce one ATP from this. So you know, kind of just make things simple. Let's just say we produce 1 ATP from all of this. Later in the cycle, we're going to produce 1 FADH₂ and then 1 NADH. So, and the thing about this cycle is that we end up back with oxaloacetate. So that's kind of the important thing to know about the cycle, is that it is a cycle. So we go back to oxaloacetate, and what that allows us to do is accept even more acetyl-CoAs and allow the cycle to continue once again. So overall, the yield of this is 3 NADHs, and then we also have 2 CO2s. We also have essentially one ATP, and we also have 1 FADH₂. So we also have 1 FADH₂ as well. However, once again, you have to remember that, well, we had 2 pyruvates, right? Meaning that we actually have 2 acetyl-CoAs, meaning that the amount that we produce is going to be double this amount. Therefore, we're going to produce 2 ATPs from this. We're going to produce 3 I can't do math, 3 NADHs here. We're going to produce 4 CO2s, and we're going to produce 2 FADHs here. So let's kind of add up everything we have so far. So we said how we had 4 NADHs inside my matrix, right? So that means that if I have 6 more, that means I have 10 NADHs in total here. 10 NADHs in total. Remember that glycolysis, that produces 2 ATP. So if I produce another 2 ATP, so far right now I have 4 ATPs. 4 ATPs. Let's see, I produce 4 CO2. And I know that for my pyruvate reaction now, I produce 2 CO2s, right? Because I have 2 pyruvates. So in total, I have 6 CO2s. Finally, for my FADH₂, I only have 2 FADH₂, because remember that I decided to take the multi-aspartate shuttle. So that would be different if I took the glycerol 3-phosphate shuttle. But yeah, essentially this is what we have so far after the Krebs cycle. All right, now after we move on to the Krebs cycle, now it's time for the electron transport chain. And the electron transport chain actually takes place in the inner sort of membrane. So it takes place on the inner membrane. All right, so while the Krebs cycle, it takes place in the matrix, we have the inner membrane. And well, this is actually a pretty cool thing that's going on. Now, the electron transport chain is going to be combined with something known as a sort of proton gradient thing that's going to go on. You're going to understand that later. But these two steps combined is going to be called something. This is something known as oxidative phosphorylation. So these two steps is called oxidative phosphorylation, and this is one way to make ATP, right? The other way to make ATP, this is something known as substrate level phosphorylation. We've seen that before. That's just when we take ADP and we add a phosphate group to it, right? And then that becomes ATP. So that we already know that. Oxidative phosphorylation, that's another way of making ATP. So these are just fancy words for different ways to make ATP. All right, let's get started with the electron transport chain. Now, with the electron transport chain, we're going to be focusing on FADH₂, and we're going to be focusing on NADH. Remember how NADH, FADH₂, all they've been doing is absorbing electrons, right? They've always been reduced, right? They've always been gaining electrons. Why? Why have they been gaining electrons? They got to put these electrons to use, right? Somehow. And there we go. This is where FADH₂, and this is where everything else, comes into play. This is where they release their electrons. And a reminder that this is actually the step of cellular respiration that actually does require oxygen. So just beware of that. All right. Everything else, the glycolysis, the pyruvate oxidation, the Krebs cycle, they do not use sort of oxygen directly. All right. They do not use oxygen, but electron transport chain does. So yeah, the Krebs cycle, it actually it doesn't need oxygen, but the thing is that when the electron, it's like a sort of compound effect, right? So the electron transport chain, right? It needs oxygen, right, for it to work. If you do not have oxygen for the electron transport chain, then the Krebs cycle will not work, because the Krebs cycle is what makes the FADH₂ and a lot of the NADHs. If they're just left there and the electron transport chain can't convert all of these things to energy, well, then it's not really going to work. So yeah, that's just an important thing to know. It's also important to know that, well, the electron transport chain, as you can see, it's definitely going to be the thing that's carrying our ATP output. Like you see, we've only produced 4 ATP so far, right? Like the electron transport chain, it's going to be carrying the ATP output. So what's going to happen first is let's talk about NADH first. NADH is going to release 2 of its protons, so it's going to become NAD+. Now these 2 protons are going to travel through these proteins. All right, so you got protein 1. There should be a protein 2 somewhere. That's not on this diagram, so I don't know why it's not on the diagram. So let's just draw protein 2. Let's just say it's right over here. This is protein 2. Then protein 3, protein 4, and so on. All right. Now take a look at what happens. So these 2 electrons are going to move from sort of area 1, or we also call this, it's called complex 1. All right. So it's going to go through, these electrons are going to go through these different complexes. And as a result, this is going to actually release energy. All right. The reason why is because each of these regions right over here, they want the electrons sort of more and more. So look, think of it as like a magnet, for example. All right. So complex 1, all right, it wants the electrons pretty badly. Then it's going to go through this Q right over here. And the Q, all that means is it's something known as ubiquinone. And ubiquinone actually carries you directly from complex 1 to complex 3. All right, so if you're NADH, you're going to start from complex 1. You're going to go to complex 3 through ubiquinone. You're going to go through the cytochrome, which is going to carry you to complex 4, and then you get out of there, okay? Now, imagine if these complexes were like magnets, and each time you move through a complex, the magnets get bigger. So the electron moves through number 1, then it goes to number 3, and then it really wants to go to number 5. So as the electrons move through here, energy is released because the electrons are sort of more attracted to the bigger magnets, right? So it's going to be more sort of stable, right? And when things are becoming more stable, they release energy, right? Think about like potential energy diagrams when, you have 2 atoms that bond together, right? When 2 atoms bond together in like the correct sort of bond length and everything like that, right? They're going to release energy as a result because they are more stable. Okay, so that's what's going to happen. Now, when that energy is released, that energy is going to be used to pump protons through. So as the electrons move through these things, the whole reason why it does that is that releases energy. And all that energy is going to be used to pump protons through. Now, these numbers are actually kind of, taking a look at these numbers right now, and it's kind of inaccurate. So if you go through complex 1 as NADH, right, you're going to pump through 4 protons. If you go through complex 3, right, because you skip directly from complex 1 to complex 3 if you're NADH, well, you're actually going to pump through 4 protons. And then finally, the last one's correct. When you reach complex 4, you pump 2 protons through it. So when you have 1 NADH, in total, you're going to pump 10 protons through. All right. You're going to pump 10 protons through. Now, you might be wondering, what about FADH₂? What about that? Well, it turns out that FADH₂ goes on a completely different route. It actually goes to complex 2. It actually starts at complex 2, right? So while NADH, it goes to complex 1, skips to complex 3, then complex 4. FADH, it goes to complex 2, then to complex 3, and then to complex 4. Now, it turns out that complex 2 actually pumps out zero protons. So yeah, that's kind of sad. And then it travels to complex 3, which pumps out 4, and then it goes to the 4th one, which pumps out 2. So every FADH₂ molecule that you have, it's actually going to pump out 6 protons, right? So for every 1 FADH₂, you're going to pump out 6 protons, okay? So yeah, there we go. Now, what's actually going to happen is we're going to have a lot of protons on this side, right? We're going to have a lot of protons on this side. A lot. Now, what does those protons want to do? I want to balance out the protons on both sides, right? It's like if you have a jar, right? Let's say if this is filled with water. This is the cool part, all right? Let's say if this is filled with water. And what you do to this water is you place some pink food coloring in there. What's the pink food coloring going to do? At first, the pink food coloring is very concentrated in one area. But what it's going to do is it's sort of going to diffuse, right? It's going to spread out to all these different areas to make the whole sort of liquid pink, right? It's going to make this into some sort of pink solution. That's exactly what the protons want to do, right? It's all concentrated in here, so it wants to find a way to get through this membrane, right? It wants to find a way through. It wants a way to get through this inner membrane, right? Because, well, it wants to diffuse, right? It wants to find a way to, restore balance, right? I want some protons here as well. So how can I go from protons in the intramembrane space to protons in the matrix? And that's where something known as ATP synthase go. This is something known as ATP synthase. This is what's going to allow the protons to diffuse and allow it to get back to the matrix. So the protons are going to go to this ATP synthase. They're going to go back, right? And as a result, ATP is actually going to be created. All right, as protons actually go through ATP synthase, it's sort of like a turbine, right? If you have like a sort of hydroelectric dam, right? When water flows through the dam, the turbine is going to start spinning. And if you took AP environmental science, right, when the turbine spins, that's going to power a generator, and that's going to generate electricity, right? So it's kind of like the same thing here. The protons are actually going to spin this thing, right? It's going to be like a sort of turbine, and that's going to generate actually ATP. That is going to generate ATP. So there we go. This is going to be essentially cellular respiration, right? The proton gradient thing that I was talking about before, that's just a fancy way of saying, okay, we have a lot of protons on this side, and these protons, they want to diffuse, right? They want to spread out to areas of lower concentration. So it's going to go through the ATP synthase, it's going to create that NADP, and boom, energy is going to be created. So you might recall once again that when you have NADH, that's going to yield 3 ATP, and FADH₂, that yields 2 ATP. This is like more older textbook sort of values. But anyways, right, we have 10 NADH, so because 3 ATP for 1 NADH, if we have 10 of them, that yields 30. And if I have 2 FADH, well, that's going to yield 2 ATPs, so that's going to be 30. That's going to be 4. Sorry, it's going to be 4, right? Because 2 times 2 gives me 4. And once again, we still have 4 ATP, because remember, the 4 ATP, oh my god, the 4 ATP came from the sort of glycolysis as well as the other steps, right? The Krebs cycle and everything. So in total, we end up with 38 ATPs, which is the maximum yield, right? But once again, this is assuming everything goes to sort of plan. A lot of times this isn't the case, right? So yeah, we get 38 ATPs out of that in total. But yeah, essentially that is it. Now we actually have to take into account one last thing. So remember how we have electrons in the electron transport chain? Well, they need to connect to something, right? The electrons are, otherwise it's going to be like a traffic jam, right? The electrons are just going to get jammed in there. Something has to accept these electrons. And it turns out that it's going to be oxygen, and that's going to be combined with hydrogen ions. So take a look. So we know that we have 10 NADHs, and each of them release 2 electrons. So that means we have 20 electrons here. And we have FADH here, so that's going to release 2 electrons as well. So there's going to be 4 electrons here, right? Because each FADH₂ releases 2 electrons, so 2 times 2 is 4. So we're going to have 24 electrons in total that we're going to deal with. Now, it turns out that when the electrons come back out, what's actually going to happen is that we're going to have oxygen. And oxygen this is going to react with hydrogen. So this is actually going to react with hydrogen ions. Now, this is going to react with hydrogen ions. And in fact, it's actually going to react with 4 hydrogen ions. Now, 4 electrons are going to be needed for this reaction, and guess what it forms? This is going to form water. This is where we get the water from. The water from the electron transport chain. All right, every time 4 electrons pretty much gets pumped out, 4 hydrogen ions are going to come neutralize it, and after that, then we're going to need oxygen. And after that, we're going to make H2O. In fact, we're going to make 2 H2O in this reaction because, there's 2 oxygens, right? And there's 2 oxygens and 2 water molecules. But there we go. This is how we make water right over here. All right. Now, if I'm using 24 electrons right over here, well, notice how I have 6 times the amount of electrons. So imagine if I multiply this whole equation by 6. right? If I have 6 times the amount of electrons and literally everything else, I'm going to produce 6 times the water. So I'm going to actually produce 12 H2O. Now you might be saying, what? But in my cellular respiration equation, right, it's C6H12O6, and this is going to react with oxygen, right, 6O2. That's going to yield 6CO2, which we saw before, right? We saw that we had 6 CO2 here, so CO2 has already been produced. But then we're going to have only 6 H2O, along with some ATP, right? So where does this 6 H2O come from, right? We produce 12, not 6, right? And my answer to your question is that this is the net water production. See, the earlier steps for glycolysis and the Krebs cycle, some of those steps actually involved water. I didn't actually say this because, simplification and everything, but some of the steps actually required water. So the net sort of gain of water is actually 6 H2O. But just know that for this step right over here, we produce 12 H2O, but the net gain is actually 6. So yeah, that is something important to know. Also note how when I multiplied this whole equation by 6, notice how I'm multiplying the amount of oxygen by 6. And notice how in my cellular respiration equation, how much O2 gas do I have? 6. So there we go, guys.